Thank you so much. I'm excited to be here today to tell all of you about Island Pharmaceuticals. If we can jump 2 slides ahead, please. So I'm David Foster, the CEO of Island Pharmaceuticals. Island is a mid-clinical stage drug development company. We're focused on repurposing strategies, and I'll explain what that means as we go throughout the talk. We're also focused on antiviral drug development. So we had an IPO last April. We went public, raising $7.5 million Aussie. The good news about that is that that's enough money to allow us to complete our phase 2 clinical trial that we're moving forward with right now. As I mentioned, we're a repurposing company, and what that means is we look for molecules that have a clinical history in one indication, a good safety profile, and then we rely on that data to move forward with a different indication. Our lead indication, as we've discussed, is mosquito-borne viruses. Our lead indication is dengue virus infection, and we have a lead program called ISLA 101. Next slide, please. So the reason we like repurposing is to get a drug developed from start to finish typically takes about 10 to 15 years. It takes about $2 billion US, and that's not what we want to do. Rather, as I mentioned, we want to focus on molecules that have a clinical history, a good safety profile, and then rely on that data to develop that drug for a different indication. Our drug, as it turns out, was initially developed as a cancer therapeutic. Years have been spent on this drug, millions and millions of dollars have been spent, but it's never been approved for anything. It has a great safety profile. It hasn't been proven to be an effective drug for other things yet. So we want to focus on the bottom right-hand side of this slide, drug repurposing, where we can eliminate the first 2/3 of this pathway and save literally hundreds of millions of dollars. Next slide, please. And the next. So what we're focused on is dengue infections. And why dengue? Dengue is a worldwide significant problem. Literally half the world's population is at risk of infection from dengue. Dengue symptoms range from flu-like symptoms, malaise, to upon second and subsequent infections, you can have dengue hemorrhagic fever. It can result in death. And there are no effective therapies. There are limited effective vaccines, and they have very narrow labels where they've been approved. So there's a significant opportunity for an orally available small molecule that might prevent or treat dengue infections. And ISLA 101 shows those characteristics preclinically. Next slide, please. So what we're also observing right now is, and we've spoken to people in endemic regions, these would be regions such as India, think of Latin America where there's endemic dengue, and what people tell us is we're fighting 2 pandemics simultaneously. We're fighting COVID-19 and we're fighting dengue infections. And it's a significant problem with the people, with the hospitals are being overrun. And again, there's just no effective therapy. Next slide. So this I thought was very interesting. This is a map showing the hotspots of dengue infection today. And as you can see, as you go from blue through yellow up to red, the incidence of dengue infection increases. So if you look at India, significant hotspot. If you look at Latin America, significant hotspot. These countries are really on fire right now with the dengue infection. But if you look at Australia, there's some regions where we have dengue infections, likewise in the United States. But on the next slide, I'll show you what it's predicted to look like in 2050. This is just 28 years from now. Now look at Australia. The northern third of Australia is predicted to be a dengue hotspot. Likewise, if you look at the United States, the southeast section of the United States also predicted to be a dengue hotspot. So it's a significant concern today. It's a growing concern. And as we'll see on the next slide, there's really no effective therapy. The numbers of infections are staggering. Hundreds of millions of people are afflicted every year with infections with these mosquito-borne viruses. The vaccines are limited. I mentioned there are some approved vaccines, but they're limited availability. Some have very narrow labels. So we think that there's a significant opportunity for a molecule, as I mentioned, an orally available small molecule that had characteristics of being able to prevent and or treat a dengue infection. Next slide, please. And the next. So let me tell you about ISLA 101, our lead molecule. I mentioned that it was initially developed as a cancer therapeutic, but it's never been approved for anything. However, based on work out of Monash University in Melbourne, Professor David Jan's lab, Professor Jans recognized that ISLA 101 was a very effective inhibitor of dengue infections in cell culture. In fact, it was effective at inhibiting infection with all 4 strains of dengue, as well as Zika and West Nile and yellow fever. Professor Jans went on to demonstrate that if you took freshly isolated human cells and tried to infect them with dengue in the presence of ISLA 101, you could block that infection. So we now have freshly isolated human cells, which are pretty close approximation to humans. We also have animal data with a lethal dengue model, as well as a Zika model where ISLA 101 is protective. With the clinical history of ISLA 101, the safety data of ISLA 101 in hand, with these preclinical data in hand, we went to the FDA and asked, can we begin a phase 2 clinical trial in dengue infected individuals, bypassing a phase 1, going straight to a phase 2 study? And the FDA said, yes, we can. So on the next slide, we'll talk about what those clinical trials are going to look like. Before we get into the clinical study, though, however, I want to tell you what we've been working on over the last several months. We've been working on our investigational new drug application, an IND. You have to file an IND and get an IND approved by the FDA before you can begin clinical trials. And an IND it's a big document. It is the compilation of lots of pieces of data. And we have many groups working on assembling this IND, and we're getting very, very close. I'm not going to go through all of the information on this slide, but suffice it to say, there are many, many pieces to this puzzle, and we're very close. One of the key pieces is getting your drug made. That's a 2-step process. The first, you have to get your active pharmaceutical ingredient, your API. Then you have to formulate that into the material that's going to be used in clinical trial. We've recently obtained our API. It is literally being shipped to our drug product manufacturer right now. So we're very excited that we're moving down this path. On the next slide, we'll see what that means for the people who are putting this program together. We've recently hired Teresa Byrne. Teresa is our VP of Clinical Operations. We're very excited to have her on board. She has years of experience running clinical trials, international clinical trials. Likewise, we're working with Larry Norder. Larry is our manufacturing expert who is really spearheading the effort to get the drug made. The clinical trial is going to be run at SUNY Upstate in Syracuse, New York. The reason for that is it's the only place you can run this study. SUNY is working on the protocol. We've engaged Camargo to work on our investigational new drug application. They're drafting it and they're in completion with that now. We've recently engaged ICON, which is the CRO that's going to oversee the clinical trials. So I think you can see we're making significant progress. We're getting close. On the next slide, we'll look at some timing and when we think we're going to be getting there. So we need to finish the manufacturing of the clinical product. We need to finalize the manufacturing section of the IND. So once the clinical product is made, we'll drop those data into the IND, finishing that last puzzle piece. We'll file the IND, and then there's a mandatory 30-day review period at the FDA. And if we don't hear from the FDA, then we can start the clinical trials. So again, we're moving down the path. Next slide, please. The clinical study, I do want to spend a few minutes or maybe a minute telling you about this clinical trial. It's incredibly exciting. What we're doing is a challenge study. And what this means is we enroll healthy individuals and then we infect them with an attenuated or weakened strain of the dengue virus. They're very powerful clinical trials. You may have heard about them. There's been conversations about these in the context of COVID-19, for instance. Notably, we're working with Walter Reed, the Army Medical Hospital in the United States, and Walter Reed recently completed a phase 1 clinical trial with the virus that we're going to use in our study. And we can tell you that what happens is people get a little bit sick. They feel a little bad. They have clinical dengue. We can measure symptoms associated with the infection, but they don't get too sick. It makes it for a very nice model to do a well-controlled clinical trial in a hospital setting. What we're going to do is a prophylactic study. So again, enroll healthy individuals, pre-treat them with ISLA 101, then infect them, and continue treating them for the next 20 days, as we've shown on this timeline. And then we want to see if we can reduce the infection compared to the control individuals. The control data is going to come from the Walter Reed study I just told you about. So we have a cooperative research and development agreement in place with Walter Reed. Under that, they're giving us all of the data out of their study. So it's an incredibly valuable piece of data. It saves us tremendous amount of time and money in our enrollment. We don't have to enroll nearly as many people as we would have otherwise. So we like partnering with Walter Reed on the study. Next slide, please. So the commercial opportunity, there's several ways we can go with this. It's pretty clear that we think there's going to be an opportunity for travelers, for the military, for people being sent into or traveling to endemic regions. We think it's going to look like the anti-malaria market, which is anticipated to be about $1 billion market here in just a few years. We also think that there may be opportunities to support national outbreaks, for instance, in government stockpiles. I've mentioned we've been speaking with people in endemic regions, and I think that there may be a real opportunity there. The final opportunity is what's called a priority review voucher, and this is the priority review voucher program is an incentive program created by the FDA in the US. And what it does is it incentivizes companies to develop drugs for underserved diseases. And the way it works is if you get your drug first approved for a disease that's on the list of diseases for which you can get a PRV, then you get the voucher. The voucher enables you to get expedited review of another program at the FDA. So you get your drug approved. If you check all the boxes, then you get this voucher, and the vouchers can be sold. They typically have been selling recently, and the last 10 sold for just north of an average of about $100 million. Just a few hours ago, there was an announcement that a company recently sold their PRV. They sold it for $110 million. So we view this as icing on the cake. It's not what we're focused on. We want to get the drug approved and get it into people to try to help people. We should check the boxes. We do check the boxes today to get a PRV. Zika, dengue, and chikungunya are on the list of diseases for which you can get one. So again, we view it as icing on the cake. Next slide, please. The team that we've assembled is second to none. The board is fantastic. Dr. Paul McClayman, significant experience running clinical trials and getting drugs approved, running companies all across the world. Anna Lavelle, Al Hansen, David Brooks, all fantastic board members. And our scientific advisory board is just second to none. All of these gentlemen have significant experience developing drugs. Next slide, please. This is a highlight of some of the benchmarks we've achieved to date and what we plan to achieve over the next year. I've spoken to most of this right now, so I'm going to pause and I will go ahead and take questions if you want to go to the next slide, please. Thank you.
Thank you, David. We'll wait as that next slide comes up. Just a question. You've only recently been listed, obviously, and there's a substantial investor notice from Jason Carroll, who's kind of increased his stake from kind of 5 to 10%. Can you give us a little bit of his background?
Well, without going into too much detail of his background, Jason is, we've spoken with him, we speak to him whenever he wants, quite frankly. And just, he's a friend of the company. He is very interested in what we're doing. He's obviously very, very supportive. And so we're grateful for all of his support.
Understood. He's clearly a fan. Now, when you were presenting, the Gates Foundation kind of sprung to mind, given their work. I think it's in malaria. Are they someone you can work with, or do you kind of continue to go down this kind of FDA support route and get that voucher? Yeah,
so the Gates Foundation is doing phenomenal work. They have done a lot of work with malaria. They're doing a lot of work looking at mosquito control. And so they're doing fantastic work. We have not spoken to them yet. They haven't seemed to have been really focused on dengue as a disease. And so we have taken a different approach, and that is, let's get this drug approved, get it into the clinic, get it approved, and move towards commercialization that way. And the appeal with dengue was the fact that it is, it spreads globally and is such large in terms of the number of people it affects. Yeah. So with ISLA 101, we had some optionality, right? We had a lot of data with dengue, Zika, West Nile, yellow fever, all of which are very bad, serious mosquito-borne viruses. But the number of people infected with dengue every year is literally orders of magnitude higher than these other viruses. It's just a significant worldwide problem. So that's where the real need is. It's a bigger market. And there's, obviously commercial opportunities associated with that, but it's the biggest problem. And moving forward, is the kind of repurposing strategy to find different drugs for different diseases? That's exactly right. So we've recently entered into, I didn't get to talk about it in this presentation. We've entered into 2 research collaboration agreements, one with Monash University, one with Griffith University. The idea there would be using technology specific to each of the universities, looking at repurposed molecules, for instance, screening libraries of molecules with clinical history against different viruses, not necessarily mosquito-borne viruses. We want to look at other viruses that are relevant to the human condition and look to repurpose molecules to rapidly get into phase 2 clinical trials.